Hello everyone, welcome back to my channel RX Essentials by Radhika. In this video, we'll be breaking down the science of immunity. So, first we'll start with founders and their core concepts. Immunity is your defense mechanism against the pathogens. Pathogens, those are disease causing agents. So first, Edward Jenner, he's the father of immunology. He developed the first vaccine against smallpox, variola. Second is Louis Pasteur, who is known for pasteurization. Third, we are talking here about is Paul Ehrlich, the father of chemotherapy. He introduced magic bullet concept and the humoral immunity concept. Fourth, Mechnikov, pioneer who discovered phagocytosis. Now, there are two types of immunity. First is the natural immunity. Let's talk about this type of immunity. So, natural immunity, which is also known as innate or inborn immunity. So, this is your non-specific, that is immediate defense, and which is affected by species or race or age. For example, if you see, yellow fever affects white races, but it usually has no effect on Negro race. Or for example, diphtheria affects children, but adults are resistant to it. Okay, so now the second type of immunity is acquired immunity. So acquired immunity is specific, that is memory based defense. So now acquired immunity is divided into two parts again, that is active and passive. So active, how you acquire this active immunity? You make the defense, for example, naturally by getting an infection or a disease like clinical or subclinical, or artificially getting a vaccine. On the other hand, how is passive immunity acquired? You receive ready-made defense. Those are antibodies, naturally from mother to the fetus, or artificially receiving ready-made antibiotics like antisera, antitoxin, immunoglobulins. Next comes our three lines of defense mechanism. First line of defense mechanism, so it's divided into two types, that is physical and physiological barrier. So in the physical barrier comes our skin and the layers of it, dermis, epidermis. Second type is the physiological barrier, which includes temperature, pH, bodily secretions, etc. Second line of defense. First is the cellular. In this we have two types of cells. First is the phagocytic cell, that is cell eating or cell engulfing method. So in this we have two types of cells, macrophage and neutrophils. So macrophages, they use pseudopodia to engulf bacteria, which is then digested into a digestive vacuole, that is lysozyme. So bacteria, they can exhibit a Brownian motion, that is a random motion using structures like cilia or flagella. Neutrophils also work through the same process. They are also phagocytic type of cells. Second comes natural killer cells, NK cells. So their main mechanism is by releasing perforins, and they are most potent against virus. So they targets virus infected and cancer cells as well, releasing perforins to punch a hole, causing the cell to swell and die. Next, cytokines. So these are chemical releasers. They releases chemicals like interferons, and they treats hepatitis, influenza or pylorama fever. So cytokines, they releases interferon chemicals which are made by infected cells, and this signals host cell to produce antiviral proteins to treat infections such as hepatitis. So there's one important point to not miss, that phagocytes move via diapedices of neutrophil through the endothelial cell to reach the infection site. Third line of defense. This line provides the most specific, powerful and lasting defense. So in third line of defense, we have humoral immunity. Okay, so this immunity is mediated by beta cells, and the origin and site of differentiation is bone marrow, and they produces antibodies. Whereas cell mediated immunity, CMI, this is mediated by T cells, where the origin of T cells is bone marrow and site of differentiation is thymus gland, and they produces lymphokine. So here we'll go in little detail with T cell types. T cells, as we have already discussed, they are the workers of CMI, cell mediated immunity. So first is a TC, that is cytotoxic cell responsible for cytotoxicity, direct killing. Second, TS, that is T suppressor cell, which is essential for regulating the response and preventing rejection in organ transplant. Third, TH, that is T helper cell, the master switch that stimulates B cells and TC cells. Fourth is a TDH, that is T delayed hypersensitivity cell, which is involved in delayed immune reactions. Okay, now let's look for B cell functions. B cell, which comes under humoral immunity. So B cell handles threats which are found outside your cells by making antibodies. So there are three steps by which B cells makes the antibodies. First step is the interaction. A B cell interacts with an antigen. Second step is the proliferation. It starts multiplying with the crucial help of helper T cells. And the third step is the differentiation. It splits into two types of specialized cells. Plasma cell, this produces specific antibodies against antigen. The second type is memory cell. This cell sticks around to provide long-term immunity. So T cell function, which comes under cell-mediated immunity. So T cell handle threats inside your cell, mainly viruses, by direct attack. So the first step is targeting. A T cell finds and binds to a virus infected cell. Second step is activation. This process activates the T cell. And the final step, that is destruction. The activated T cell then releases a toxin granule to destroy the infected cell, stopping the virus from spreading. So that's the entire specific immune response. B cell neutralize and T cell directly kills. So that's all for today's topic, Immunology Simplified. If you like the video, don't forget to like, share and subscribe.